So I'd like to welcome everyone here for this event. This is exactly the sorts of things that the Genome Center does and should be doing, hosting events like this, having scientific conferences and other events that really bring the community together in New York. So for those of you who are not familiar with the Genome Center, I'm going to give a brief series of slides to quickly tell you about us. We've been in the space for about 6 years, and we were founded at that time with the cooperation and contribution of our 12 partner institutions, which is on the first slide here. So you can see these are the institutions that came together over 6 years ago to found the Genome Center as to provide the infrastructure and intellectual core of genomics in medicine in New York City. And you can see basically every university, medical school in New York is represented, in addition to ones that you probably wonder about, the Jackson Laboratory, which makes wonderful mouse models, and they've been part of the Genome Center from the beginning. So we're focused on advancing genomic disease-focused research that leads to scientific advances and therapies for patients in neurodegenerative disease, neuropsychiatric disease, cancer, and we leverage our strengths in whole genome sequencing, genomic analysis, and the development of genomic tools. So tonight you'll hear a sample of that, which really spans these different areas, and I think will provide you a glimpse of the exciting both technology work here that's being done, as well as basic research. We have staff scientists as well as core faculty members with the latter leading independent research programs and engaging in research that supports both the New York Genome Center and the wider community. And in fact, all of our faculty hold joint appointments at our partner institutions. These are tenure track appointments that all of our faculty hold. So as I said, a central part of our mission is to bring the communities together and being a nexus for collaboration in genomic research. We bring together our faculty, scientists, clinicians of our member institutions, all around the mission of being the leader in genomics. So let me give you a few highlights. So this just summarizes very briefly the things we do. We have high quality whole genome sequencing at scale. And I should say that that's an area that we've really excelled in. We are one of the first of the genome centers to make a commitment to whole genome sequencing at large scale. And as I'll mention later, we have sequenced 22,000 whole genomes for autism. I mentioned the computational expertise we have. We've got a great computational biology group that works both on projects within the Genome Center and in collaboration with our partners. We have the capacity now to sequence 40,000 genomes per year. We have received numerous awards in publications, some of which I'll mention when I introduce our faculty. And we have been parts of partnerships and consortia, a couple that I'll mention, and that we are very successful at bringing in research grants both from NIH and from private foundations to fund the research here. So we recently have reconfigured our strategic plan. We used to work on many different disease areas and have a fee-for-service structure. We don't do that anymore. We're really focused on research. We are a research institute, and we're focusing in 3 disease areas: neuropsychiatric, neurodegenerative disease, and cancer, and I'll briefly mention that. I won't go through this in detail, but just point out that we have both internal faculty, as I mentioned, that have joint appointments, but we have associate faculty who actually don't have laboratories here. Some of the senior faculty, like Mike Wigler, Cold Spring Harbor, Harold Varmus at Weill Cornell, Simon Tavre, who recently joined the Columbia faculty, who used to be the director of Cancer Research UK, Greg Hannon, Evan Eichler, and myself. And so I'm here full-time, but the other people are on committees, they're on joint grants and so on, and they provide external senior scientific leadership for the Genome Center. And as you can see, we have a sizable junior faculty, and these individuals, as I mentioned, have joint appointments, and we're extraordinarily proud of our faculty. We are able to recruit at a very critical time an extraordinary group of individuals. We recently opened our program to the community through having an affiliate membership program. We now have 65 affiliate members that are part of the Genome Center. And we have a number of disease working groups that these affiliates are part of. It's a Genome Center Cancer Group, which is led by Harold Varmus and Charles Sawyers. Computational Cancer Genomics, which is being led by Simon Tavre, who I mentioned, and Saurabh Shah, who is a new faculty member at MSKS. We have a Neurodegenerative Disease Working Group with Alison Goat, who recently joined Mount Sinai faculty, Himali Fatnani, who's a faculty member here, and Phil DeYager, who recently moved from Harvard to Columbia. I should say one of the roles that we've played in the city is that we have helped recruit a large number of both junior and senior faculty to the city, to the institutions here, which I think has been a big part of our contributions to the community here. We have a neuropsychiatric disease working group, which we're just putting together, and a population genomics working group, which is led by Adam Siepel, who runs an integrated biology program at Cold Spring Harbor, and Emer Kinney, who is a population geneticist at Mount Sinai. So in each of these groups, we're now recruiting scientific directors in each area. And the plan is that we will work together to find new directions, write grants together, and really sort of enable the community in these critical disease areas. This just shows an outline of the Center for Genomics of Neurodegenerative Diseases, which is led by Himali Fatnani. And it's a consortium of 29 member institutions in 5 countries. We raised funds both through philanthropy and to the ALS Association to fund whole genome sequencing and RNA sequencing for ALS patients. And this was designed on a completely open source configuration so that all the members submit their samples, the sequencing is done, they have immediate access to the data. And we have a semi-annual meeting to discuss the work and work on the analysis of the data. One of the strong programs here, which is funded by a center grant from the NHGRI at NIH, is an autism. And these are the co-PIs on the grant. It's Mike Wigler, Evan Eichler, Mike Sodi, who is heads the computational group here at the Genome Center. And we have sequences, it says there, over 22,000 autism whole genomes and we're doing RNA sequence on transformed blood cells from all these individuals. And we believe that we are really configuring an approach to whole genome analysis that no one else is really doing to understand the nature of sequence variants in whole genome in introns and in non-coding sequences that are related to disease. We are going to have a discussion later by by Nicolas Robine on the Polyethnic 1000 project, which came out of our GCCG. I won't go into this in detail because he'll cover that, but it's a great project that's brought all of the representatives of the cancer community in New York together to find a way to provide patients from underrepresented communities in New York with state-of-the-art cancer genomic tests. This just demonstrates one of the central features of this program, of this P1000, and demonstrates the power of doing genomics in New York City. What it shows here, this colored map is quite amazing. As you can see, block by block in neighborhoods, where you can see Black, Hispanic, Asian, white. You see Southern Caribbean. It's an amazing map that really indicates the diversity and location of ethnic populations in New York. And as you probably know, this is one of the major issues in genomics today of really including ethnic diversity within analysis, because a sequence change in an individual from one ethnic group may or may not have the same consequence with regards to health in another ethnic group. And so it's a key issue in New York City and beyond. We have a clinical laboratory, a so-called CLIA lab that is licensed by the state of New York to provide tests. And you'll see we have the tests here, which include constitutional whole genome, which is genetics of whole genomes for genetic diseases, for rare disease and predisposition for conditional approval. We have whole exome, targeted variants, and the oncology exome and constitutional SMAS, which is a copy number variant assay. And then finally, we recently provided the first and only cancer whole genome approval in this fall. So New York Genome Center is the only institution in New York or beyond that can do whole genome genetic tests on cancer. So we've built both the scientific base, the computation, the infrastructure to really do clinical genetics, and that's a very significant focus of what we do. So I am going to introduce the first speaker. Dan Landau is an MD, PhD. He's a physician scientist who has pioneered the study of cancer evolution as a central obstacle to the path of curing cancer. His work has delineated evolutionary course that allows cancer cells to rescind the multicellular contract, diversify, and become resistant to cancer therapy. In 2016, Dan was jointly appointed as an assistant professor of medicine and a member of the Institute of Computational Biomedicine at Weill Cornell Medical School, and is a core member of the faculty at the New York Genome Center. He cares for bone marrow transplant patients at the New York Presbyterian Hospital. His recent awards include a 2018 NIH Director's New Innovator Award, the inaugural Aspire Accelerating Scientific Platforms for Innovative Research Award from the Mark Foundation for Cancer Research, a Stand Up to Cancer Philip Sharp Innovation Award and Collaboration Award, and the Pershing Square Sohn Prize for Young Investigator in Cancer Research and Lung Cancer Discovery. And you'll see from his talk today that he's really pursuing the application and development, I should say, of state-of-the-art genomic tools for both mixed populations of cancer cells as well as single-cell analysis. So without further ado, Dan.
So I decided to kind of focus on one project that I think represents what we do in the lab, how we think about integrating between a clinical challenge, a biological challenge, computational innovation, molecular biology innovation, and bringing all of this to new tools that we can apply in the clinic. So our work has shown, and the work of others, that although we have good methods to treat cancer, good drugs, new drugs, exciting drugs, good surgical approaches, radiation, and so forth, cancer is also able to evolve, to develop resistance, to recur. And one of the challenges that we face is how are we able to monitor cancer in a dynamic fashion, in a real-time fashion. That's a significant challenge. And think about any engineering process that you're aware of, a successful engineering process. Most of them are based on this unifying principle of real-time monitoring and optimization. Think about the air conditioning in this room. It's a little chilly, but in principle, the air conditioning system is able to continuously monitor the temperature and apply some adjustments based on that. And that's true to many successful engineering processes that we have anywhere. Now, in contrast, when we look at cancer, sometimes there's a really big gap. So for example, as a clinician, a patient could come to me after curative surgery, for example, for lung cancer or for melanoma. The surgeon tells the patients, I took everything out. The radiologist tells the patients, I can't see anything there. But as an oncologist, we know that there's a substantial proportion of these patients that actually are going to have recurring disease, that are actually going to have disease. But there's nothing that I can tell the patients, well, I'll do a test and see if there's something left or not, because all of our systems for monitoring are just failing the patients at that juncture. The only readout that I will have for this patient is whether they will come back to me into the clinic maybe 2 years later with a recurrent disease, metastatic disease, high burden, where de facto we have failed the patient. So that's a challenge. And not only that, but because we recognize that there is a substantial proportion of patients that actually will recur, we give something that's called adjuvant therapy. Adjuvant chemotherapy, now adjuvant immunotherapy. So that individual that had surgery, the surgeon is telling me that nothing is left behind, the radiologist is telling me nothing is left behind, will now go on therapy for 6 months or for a year, sometimes very aggressive therapies, very toxic therapies, very expensive therapies. And throughout the entire process, there's nothing I can tell the patient, look, it's working, or it's not working, or maybe we should try something else, because we just lack the monitoring tools for real-time therapeutic optimization. So for example, there's an intervention here, maybe it's surgery, maybe it's therapy, but there's nothing to tell me whether this patient maybe needs a different therapy, or maybe this is that patient and maybe they're responding well and maybe they don't need further therapy and so forth. Now, one of the exciting developments of recent years, it was discovered that in the blood, in the peripheral blood, small fragments of the DNA are shed into the peripheral blood. So even if the tumor is in the lung or in the gut or in the brain, small parts, small fragments of the DNA of that tumor are actually shed into the peripheral blood. So imagine a large pool, and somewhere along the periphery of that pool, there is a leaky faucet that's dripping, parts of that DNA into a large pool of the blood. And that's exciting because for monitoring purposes, that allows us to go back to the patient and draw a blood sample every 2 weeks or every month. And that's a much easier way to monitor the disease rather than take the patient to the OR and see if there's disease there, the operating room. And indeed, this is so exciting that there's a large swath of both academic research and industry that has been really geared towards optimizing this method. And what the majority of all of this research, what it has done is that it tries to tackle the problem of how do we find these fragments if they're so rare, right? Think about that faucet leaking along that pool. How do I find these rare fragments out of the entire pool of fragments that are coming from the blood? And essentially 99% of the community decided that the best approach is to try to find individual sites in the genome, let's say a cancer gene, and then sequence it very, very deeply with very sophisticated methods, such for that individual part in the genome, you can find even one particle out of 1,000,000, right? Because that would allow you the sensitivity to tackle, to find these rare populations and so on. And that's exciting, but in practice, even though, and you're MIT crowd, so I'm allowing myself to use numbers and so forth, but even though we have a method that can go to 1 to 1,000,000, when you look at the data, there's a disconnect. And the disconnect is that actually the sensitivity stops at 1 to 1,000. So how come if you have a method that's sensitive enough to go to 1 to 1,000,000, there's a hard stop at 1 to 1,000? We know it's not the molecular biology. The molecular biology works. When we mix things in the lab, it works. So why is there this disconnect? And the disconnect comes from something that is now recognized, that in fact, although your method can go to 1 to 1,000,000, there's only very few fragments in that peripheral blood sample. I only take a few mLs of blood, and there's just not enough fragments covering that site. So imagine that this is the large stack of 1,000,000 different fragments. I could potentially detect just one of them if it was there, a very small dilution, and yet there's a ceiling, a boundary. I was only given 1,000 fragments. So that puts an upper limit to my sensitivity, or a lower limit. I always confuse the two. So I will never be able to go below that, even if my molecular biology is ideal. There's a disconnect. I have a method that can take me to 1 to 1,000,000, but because I'm going in one particular site very, very deeply, and there is this ceiling, we are not able to detect cancer in many of those instances where we need it the most, where the disease burden is lower. So we thought that we have to rethink the problem. We have to rethink how we do this. And essentially, we kind of flip the problem on its head. Because from the statistical perspective, looking at one site very, very deeply, maybe like across 100,000 fragments, from the statistical perspective, it's the same problem as looking at 100,000 sites only with one fragment. So we flip the problem around. In a sense, instead of looking at one site with these tall stacks, we reason that we can get the same statistical power by having a broad sequencing of the entire genome, going for the tens of thousands of mutations that are found in these tumors. So essentially, we're not limited by this limit, the ceiling of how many fragments are there. So again, this is a different perspective. If the tumor is shedding parts of the DNA, instead of just looking at these particular sites and crashing our head again, bumping our head against the ceiling of the number of genomic equivalents, the number of fragments that is limiting our sensitivity, we can go more broadly across the entire genome and kind of flip the problem on its head. And what that does mathematically, as you can imagine, is it really transforms the detection landscape. Instead of having essentially a detection sensitivity that precipitously declines once you go below that 1 to 1,000 threshold, now you have a detection landscape that allows us to go after much lower frequencies of tumor. Of course, this required significant computational innovation because we're going after those very rare reads, fragments of DNA. We have to, in a sea of many, imagine that little faucet in the pool, we had to develop machine learning methods that are able to go through those fragments one by one and tell us when do those fragments are coming from, let's say, a sequencing artifact or some noise, or where they're coming from true signal. We actually presented this in leading machine learning conference ICLR, and this is available online. And a long story short, we show that with this method, instead of stopping at 1 to 1,000, now we can engage with sensitivity that is 2 orders of magnitude lower, down to 1 to 100,000. So how does it work in practice? So we take a tumor from a patient, We identify their different types of mutations in that particular tumor. That's a fingerprint for that individual patient. And then we go into the peripheral blood, into the cell-free DNA, into the plasma, and scan the genome for traces of that signal, for traces of that fingerprint. And the hope that they will be able to tell the clinician and the patient, look, there is disease left. We should do something more. We should give more therapy. Or perhaps, no, there's no disease left. We should just monitor. So how does it work in practice? So we looked for lung adenocarcinoma, which is a difficult model for established methods because the frequency, that faucet, is actually leaking a very small amount. And we looked for very early disease. You see a lot of stage 1, even stage 1A. And we show in essentially all of the cases that we look at, and we're able to detect disease even in those early stages because of this radically higher sensitivity. We don't detect these signals in normal individuals. And when we say normal, our normals are not, college-age students, but rather individuals who go to our clinicians for example, tobacco-associated screening programs. So those are older individuals with exposures to carcinogens and so forth, and yet they don't show those signals. So this is a highly sensitive method that still preserves its specificity. But I told you that the ambition here is to go beyond detecting cancer that the radiologist can see on a CAT scan. Our ambition is to be able to tell the clinician and the patient, even after both the surgeon and the radiologist say that there's nothing left behind. And as you see here, we take patients after surgery. So this is 2 weeks after the surgeon took everything out. And we see that in some individual, we do have residual disease detectable by this method. And those individuals tend to have adverse clinical outcome. So those are more likely to recur. And again, this real-time information potentially, and this has to be confirmed in clinical trials, which we're working on, would be able to help the clinicians and the patient navigate this black box where we don't have information to optimize therapy. So just to summarize, we kind of rethought the problem. Instead of doing this deep targeted sequencing that has this ceiling on it of detection, we kind of flip the problem on its head, and now we try to find tens of thousands of mutations that are found in these tumors using broad, shallow sequencing. We had to develop the computational tools to do so because the computational tools were just not there because nobody tried to do something in this fashion. So we had to develop machine learning methods that allow us to scan through those reads and identify the ones that bear cancer-related mutations. And through this genome-wide integration approach, now we can deliver potentially 100 times greater sensitivity and allow us to start tackling a major unmet need of how do we guide clinicians and patients when the disease burden is low. That's where we need this information because we think when the disease burden is low, our interventions are most efficacious, right? And yet we don't have the information to guide interventions. For example, again, melanoma, surgery, the patient doesn't have any disease, but now we realize that we have to give immunotherapy, and you'll hear more about it from Neville, immunotherapy even in the adjuvant setting. But how do we know it's working? Those are highly toxic and expensive therapies. How can we tell the patient, look, the disease is going away or it's not going away? Currently, the only readout would be 2 years later if the patient comes back with metastatic disease. So we aim to change that. And obviously, one of the kind of interesting aspects that related to a place like the New York Genome Center is that we're very committed to entrepreneurship and also to bringing these type of transformative tools to the bedside. And we realize that the ability of an academic lab to do this is limited. We're limited to do this effectively. We're better on the R&D, we're better on cutting edge technologies, and what we need is to form or ideally spin off a commercial entity that would be able to exploit this new technology and to bring it to the bedside as soon as possible, and we're working on commercialization of this and engaging funders and so forth. I'd just like to thank the many people who worked on this. In my group, Asaf Ali led it with a bunch of brilliant computer scientists. We had a lot of help from the Weill Cornell group. Nasser Turki is a leading surgeon there. Nicolas, who you're going to hear from, and his group have been helping with some of the computational biology aspects. And we've also worked closely with the New York Genome Center Innovation Lab, and that's a unique aspect of this place, that it has a dedicated group that is working on developing new molecular biology technologies. And it's been a privilege to work with them, and you'll hear from Eleni, who represents that group as well. We're grateful for our funders, and thank you for your attention.
Dan's talk prompted another point that I forgot to make, and that is that the third floor of this building is part of the Genome Center. And we were able to partner with the state of New York a year and a half ago to renovate the space for a biotech incubator, which is called J-Labs. You may have heard that Johnson & Johnson has a series of these incubators throughout the world. They're in London, they're in San Francisco, San Diego, and we have one on the third floor here. It went up very quickly, and it's had a huge impact on the city. In fact, it was really the first real biotech incubator in the city. On the third floor here, I think there are about 25 companies there now with 2 or 3 people looking for ways to start their companies. So I'd like to introduce the next speaker, who's Neville Sanjana, who is a core faculty member at the Genome Center. He's also an assistant professor of biology at New York University and assistant professor of neuroscience and physiology at the New York School of Medicine. Neville is a MIT alumnus and holds a PhD in brain and cognitive sciences from MIT. So Neville is a bioengineer. He creates new tools to understand the impact of genetic changes on the nervous system and on cancer evolution. His lab has harnessed high-throughput gene editing to pinpoint which regions of the genome are involved in diverse diseases. His recent awards include being named the Leichtung Family Investigator by the Brain and Behavior Research Foundation that supports his work in autism and functional genomics, the AAAS Wachtel Prize for Cancer Research, the DARPA Young Faculty Award, and the NIH's New Innovator Award in 2017. So he's another example of the exceptional faculty we have here who really are excelling in a national and international way. And you'll see from Neville's talk that he's doing amazing creative things to do whole genome scanning using the CRISPR technology, which I think he's one of the best in the world at doing.
So thanks everyone for coming. Thanks, Tom, for that overly generous introduction. And so, so I'm going to tell you a little bit, continuing with the cancer theme tonight, which I think is going to carry through to at least one more talk. I'm going to tell you a little bit about our work in the lab over the last few years to use gene editing tools like CRISPR, which was just mentioned, which is one of the enzymes pictured here, to discover predictors of cancer immunotherapy response. We just heard this really nice talk from Dan about this need for continuous monitoring to understand who responds to a particular drug or not. And one other thing that we're very interested in doing is to say in advance, before we even treat somebody, can we figure out whether this person is a good candidate, especially when some of these therapies are extremely expensive, up to now $500,000 with some of these newest immunotherapies for blood cancers. So before I dive into that, I just want to get us all on the same page, talk about, when I talk about genome engineering or gene editing, what do I mean? I think the natural analogy before you think about the genome or about DNA is to think about all the ways we currently access and store information, right? We've got beautiful libraries, we have Wikipedia, we have programmatic ways to manipulate information like making a web page or writing a piece of code in R that does something. And we've been doing this for many, many years. You know, it's from a personal computer to an iPhone. But it's not really been the same with the genome is the thing. That with code and with computers, it's been terribly advanced over the last 30, 40, 50, 60 years. But with DNA, until recently, we've had very few tools for easily manipulating it. There's pioneers in this field that have been doing it for quite a while, including Tom, who's famous for writing one of the first manuals for how to manipulate DNA. And it's important because DNA is the language of all living things. It's the code that runs us. And so it makes sense that we would desire some kind of tool set to be able to manipulate this code, to be able to make changes to it. And so how do we do this when we talk about genome editing or genome engineering in the lab? So what it comes down to at its essence, when we're talking about complex genomes like a human genome, 3 billion A's, T's, C's, and G's, is to take advantage of actually the natural gene repair mechanisms, the things that prevent us from getting cancer every day when we walk around with UV radiation or other carcinogens in the environment. So to take advantage of natural gene repair mechanisms to introduce novel genetic sequences. We want to trigger those natural gene repair mechanisms, but to do our own bidding. And so I'm showing you one example of this, which is stealing a protein that's naturally found in jellyfish, those jellyfish that you can find off the coast of Long Island here. You can shake them up and see them, that they glow green at night. So we can take these and put it into an organism that doesn't normally glow green, like mice. And so this is the idea of triggering some genome repair in the germ cells of these mice so that we can integrate in this jellyfish gene into the mouse. And so I think a nice, this illustration was done for one of our papers when I was in Feng Zhang's group at the Broad. This is a very, I think, apt illustration. So we're trying to use something like a molecular scissors is a nice way to think about it, to make breaks in the DNA and to trigger those gene repair mechanisms. So what triggers them? Making breaks in DNA. And the scissors that we use, that I'm sure almost everyone here has heard of at the Enterprise Farm, unless you've been living under a rock for a few years. It's been in many different kind of well-known journals like Science, is this tool called CRISPR. And I don't have time in 15 minutes to give you the full history, but suffice it to say that over the last 5 years, it's had a tremendous impact on biomedical science broadly, not just on genome engineering labs like mine, who like to play with this stuff just for the heck of it, but for people studying many, many different diseases, not just cancer, not just neurodevelopmental disorders. And these guys function essentially as those scissors that when they make a cut in DNA, they trigger that endogenous gene repair mechanisms. And I really like this quote from Hank Greeley, which I stole from a New Yorker article, which kind of sums it up very nicely. The Model T wasn't the first car, but it changed the way we drive, work, and live. So CRISPR has made a difficult process cheap and reliable. So people have been doing this, as I alluded to, for decades, been able to manipulate DNA to greater and lesser extents. But what really has been the game changer with CRISPR is that it's democratized things. It's made it so that really almost any lab or a group of scientists can do this quite easily now. Okay, so I wanted to set the stage a little bit about the technology, since I thought MIT should be a kind of technology-oriented crowd. Now I'm going to tell you a little bit about the biology, our kind of biological problem that we're interested in. And the work I'm going to tell you about is a collaboration with two very talented scientists, Shashank Patel and Nick Restifo at NCI. And these folks have been working on immunotherapy for quite a long time, and I was just privileged to join them and apply some of our genome editing tools to these problems that they introduced me to. Again, another technology that you've likely heard of, as it received the Nobel Prize just a few months ago, is cancer immunotherapy, the idea to harness the immune system to fight cancer. And this has just, over the last decade, when it's really entered the clinic, immunotherapy, it has just reverberated through the cancer world in the sense that in cancers that are very advanced, where we normally don't have much to offer the patient, we've seen things in some minority of patients. We've seen long-lasting remission of cancer, which is really amazing. In the cancer that I work on the most, melanoma, it's really dramatic in some. You meet some of these patients and their lives have been changed for now several years. Because it's an immune treatment, it can be long-lasting. Your immune system keeps memories of all the diseases you've had, and in that sense, if you have a response, basically you can fight off this disease for a long time. If it comes back later, your immune system remembers it. It's very specific compared to the things that Dan was talking about, things that have horrible side effects like chemotherapy or radiotherapy, because they attack both cancer cells and healthy cells. Immunotherapy is rather specific. It has some side effects rather specific for cancer cells. And because it uses your own immune system, it actually can be quite a universal therapy, not targeted to maybe perhaps one specific cancer, but useful in many different cancers. And for this, all these reasons and many more, the folks who now 30 years ago did the initial work in some mouse models were recognized with this prize. Let's see here. So that's the rosy picture. The less rosy picture is that it doesn't work for everybody, and we've known it for a long time. We've known it for more than 2 decades, that there are certain genes, but when you lose the function of these genes, these patients become resistant to immunotherapy. And that's true whether they're receiving things like adoptive T-cell therapy, which is this is something a study Nick did quite a while ago, or some of the newer things like the checkpoint blockade therapies that are now the kind of FDA-approved immunotherapy that you hear about regularly in the news, like anti-PD-1 therapy. And you see, even in these 2 different journal articles, 20 years apart, you actually see some of the same gene names cropping up, the same mechanisms of resistance cropping up. And so we wanted to apply genome editing tools to kind of figure out broadly, what are all genetic mechanisms of resistance to immunotherapy? And the idea is quite simple at its heart. So I explained CRISPR as a set of molecular scissors. So now I'm going to tell you about applying those scissors into tumor cells, and to be able to manipulate the genomes of tumor cells, to be able to remove the function of certain genes and then test them with immunotherapy, something like a T cell that's engineered that would normally kill that tumor cell. But can we do something to change one of those genes out of the 20,000 genes in the human genome to make the cell now resistant to the immunotherapy? Because that'll give us a clue as to what mutations, when we see them in patients, we should be worried about before treating with immunotherapy. And it's hard to overstate how difficult this problem is. Because the code that runs you is big, it's 3 billion letters, and because the number of genes, forget about all the code, just think about the genes, which is 1.5% of that, that's 20,000 genes. This is hard stuff to do in the lab if you're going after it one gene at a time. And so the approach that we developed and have been using in a whole wide variety of biological problems, including immunotherapy resistance, is to scale up CRISPR. Because CRISPR is such a programmable system, instead of going after one gene at a time, how can we scale it up to achieve something like genome scale? And so here, I just have this sentence here from On the Origin of Species from Charles Darwin. What's commonly done with CRISPR is we go after a certain gene. If we think of each of these words as a different gene in the genome, the entire book is the full genome, we can take out that one certain gene. But what we've been able to do is develop libraries, large libraries of CRISPR reagents, where instead of taking out one gene in the genome, we just go through every single one of those 20,000 genes and take them out in a massively parallel fashion. And what we can do is create a pool of cells that each have different genetic manipulations, different genes modified, different genes removed or overexpressed if we'd like, and to use something about the phenotype here, resistance to cancer immunotherapy, resistance to a T cell coming and killing that tumor cell, to guide us, phenotypically guide us to the right answer. It's this gene is important for this cancer phenotype, this drug resistance phenotype. And so that's kind of the high level idea. Here's the diagram that shows you how the sausage is made. And this is the part I was just talking about, to have a pool of human, in this case, melanoma cells, where each cell has a different one of those 20,000 genes in the genome removed. And to treat it with engineered T cells, very similar to the approved therapies that are now out there, and to understand which particular genetic manipulations are resistant to this T cell killing. And as Dan said, it's an MIT audience, so I can show some primary data. This is some of the primary data from the screen. And what we were really pleased to see, if you just focus on this top right corner, is the top 2 hits from our screen were very, very well-known escape mechanisms of immunotherapy, very well-known mechanisms previously of people who are resistant to immunotherapy, unfortunately for which immunotherapy fails the patient. And these proteins are not just two random proteins. They're actually the two proteins that come together to present foreign antigens on the cell surface. So when the T cell is making its rounds as a security guard and doing surveillance over your entire body, this is who tells it, hey, there's something wrong with this cell. And again, if you don't have any antigen presentation, it's very hard for that T cell to understand. In fact, it basically completely abrogates the T cell response. And so to see these as our top two resistance mechanisms, this is not novel biology. It had been known for decades. But for us, it was a really good positive sign. And again, I'm not able to go into all the details, but we were happy to find actually many, many new genes that we were able to look at across different melanomas, across different cancers that weren't skin cancer, and even using different kind of T-cell cancer antigen pairs, looking at it both in human and mouse models. I just show you one example so you can see what discovery looks like here. This is a receptor that no one had any idea that does something with cancer immunotherapy. And what we did is we used CRISPR, we knocked out this particular gene. This is just one gene we had discovered. We had never heard of this gene before, out of the 20,000 genes in the genome, and we put it in vivo into a mouse. Mouse grows a tumor with these cells that are missing this gene. And what we now see, just to cut to the chase here, is that if you knock out this gene, it really impacts survival. It makes a tumor that's normally well treated by immunotherapy much, much more poorly responsive to immunotherapy. Similar, but not quite as bad as knocking out that complete antigen presentation system that we knew was very, very bad. To get a sense of the precision medicine side, I kind of hinted at this, the last thing I want to show you, is that we were very interested because we discovered a lot of new genes that people hadn't implicated in cancer immunotherapy response. We wanted to say, well, we made all this in the lab, right? We mutated these genes in a dish. Do you ever see this in actual patients? So we took the data that was out there. We took these 3 studies with about 200 patients. And these were studies where they had some sequencing of the tumors, and they had followed these patients longitudinally to see whether did they respond to immunotherapy or did they relapse. And what we found is that if you look at the genes that are pulled out by this what we call the 2-cell type CRISPR screen, meaning tumor cells and T cells, and you look at the ones that are kind of not way down the list, but the ones that are the top hits, we really find that these genes are enriched for loss of function mutations, specifically in non-responders, suggesting that this assay and the genes that are discovered has a predictive value. It lets us deliver a little bit, at least, on this idea of precision medicine, that we can start to say, hey, maybe Novartis, we shouldn't spend $500,000 treating this person with Novartis's Chimera treatment because they're likely to not respond. We should look for a different treatment for this person. And that's what this slide says here. We're just scratching the surface here with this. And we have several projects in the lab ongoing to do a better job of this, both the computational side and also on the experimental side. So I just talked about cancer immunotherapy, but the idea of being able to manipulate every gene in the genome and identify disease-relevant genes is something that really can apply to any disease where you have some relevant selection, right? In neural disease, you can look at synaptic dysfunction, you could look at neurodegeneration. And these are some of the few other areas we've applied this to that I can't talk about today, but you can see there's a lot of work in the lab in cancer. And these are the wonderful folks that I get just delighted to go in every day and hang out with, some of the collaborators on our work, and of course the people who funded it. So thank you.
In addition to our faculty, we have an outstanding scientific staff. And you'll hear from 2 of these staff members for the rest of the program. So Nicolas Robine is Assistant Director of the Computational Biology at York Genome Center. He was actually one of the original employees of the Genome Center way back when we started. He has played an absolutely key role in cancer genomics, that is the mathematics of trying to understand the nature and effects of sequence variants in genes that are related to cancer. And he leads a interdisciplinary team and staff scientists and faculty, as well as research collaborators to explore and test genomic tools and ideas. So he He's in charge of the RNA-seq analysis and cancer genomics within the computational biology lab at the Genome Center. And he is going to talk today about the program that I referred to earlier called the P1000, which I'll let him talk about. But as I said, it's an exciting program that we believe that we're both going to learn things about the ethnic basis of mutation in cancer, and also be able to equip community hospitals with the ability to do state-of-the-art genetic tests. And so this has both a societal impact and research impact as well. Nico?
Hi, everybody. So one of the small goals that we set for ourselves at the New York Genome Center is to apply cancer genomic and cancer testing to every patient. And so we could imagine one day every patient, at least in New York, being offered cancer genomics testing. Currently, it's going in some of the hospitals, but we would like for every patient to be offered that possibility. So the project I will talk to you about is called Polyethnic 1000, and the goal is to advance cancer genomics by studying ethnically diverse and underserved patient population in New York City. So one problem we have in cancer is health disparity. And so among those different aspects of disparity, one of them is racial, ethnicity. So we see here for incidence of cancer, if we take all of the cancers, we see differences between white patients population, non-Hispanic black patients, Asian, Pacific Islander population, and the Hispanic population. These 4 bars here are for the male. This is for female. And so if we go into detail for colon cancer, we also observe some differences in its incidence of cancer. If we go for breast cancer, we also observe a difference. Now, if we go for mortality, we also observe difference between those 4 categories that we identify here. This is all based on self-declaration. So when the patient comes to the hospital, they have to check a box, which race do they, or which ethnicity do they declare, do they associate themselves with? And we observe that if we take all of the cancer, non-Hispanic black have actually a higher mortality than non-Hispanic white. So a little bit more incidence and more mortality. And we observe differences in mortality for male, for female, in colon cancer, in breast cancer, and basically in every cancer. So what are the source of those disparity? One of them is socioeconomical factor. You can imagine cultural factor. You can imagine diet. You can imagine incidence of smoking having an effect on the incidence of lung cancer, for instance. But we also know that there are some more molecular aspects of this disparity. Drug metabolism is known to be different between different ethnicities. Microbiome, the type of bacteria that we have on our body, can be different and can have an effect on cancer. It can be different between different ethnicities. Tumor microenvironment, the way a tumor presents in the pathology group, in the pathology department, when we cut slides, when we observe the type of cells, the shape of the cells, there can be differences between the different ethnicities. The driver gene mutation is something we will talk about, but also predisposition genes. So we talked about BRCA, that's a gene that predisposed for cancer, and we know that the occurrence of those predisposition genes can differ between different ethnicities. So all of those aspects interplay together, and what we are going to look at is really at the genomic level, at the molecular level, what kind of differences can we observe? So one example here of difference is predisposition genes. So that's summarize here the test of certain genes that are known to be associated for breast cancer and ovarian cancer. And those are 4 categories: white population, Ashkenazi ancestry, Hispanic, and Asians. You see that the 2 main genes that we see here are BRCA1 and BRCA2, and they account for maybe 40% of the patients in these white populations are carrier for mutations in those 2 genes. If we take the Ashkenazi ancestry, it's more like almost 60% of the population. And actually, that's in that population that those genes were discovered. If we take the Asian here population, that's even more. That's probably close to 70% are carrier either BRCA1 or BRCA2. And we see for the other gene, there are differences. I think there are P10 is present here in the Asian population, but we don't see it in the Hispanic population, for instance. So we see that there are differences in the proportion of the population that carry those genes that are associated with the development of those diseases. Now, if we look at somatic mutations, the mutations that are acquired in the tumors, this is an example in lung cancer where we see that in the Asian population, about 50% of the patients with lung cancer are carrier of a mutation in the gene called EGFR. And in the European population, that's about 5 to 15%. That's a dramatic difference. That's based on a very large number of samples that have been tested, and there are clearly a huge difference in the number of patients that carry those mutations. So that's what we are going to look at. So I prepared a slide on cancer genomics just to give you an explanation of what we're doing when we say we will do cancer genomics. So this is a representation of normal cells, and very typically we take blood cells that, except in the case that Dan talked to you about, don't carry somatic mutations. They carry mostly the germline genome. We can extract the DNA, here it is, and we can sequence it on one of our fancy sequencers here. We take all of the small reads, we align it to the genome, and that's what it looks like. That's really the standard way to look at the genome. Is here. We see all of the reads carry a small sequence. They align to the genome, and we can study this thing. We take then the tumor cells that are here represented in purple. We extract the DNA, and so every time we have a biopsy, we have a mixture of tumor cells and normal cells. That's why I put also some yellow cells here. We extract the DNA, we sequence, we align to the genome, and we see that we can have a difference. Here, that would be a difference. That would be a mutation that is present only in the tumors but not in the normal. So by comparing the profile of the alignment in the normal genome and in the tumor genome, we can identify all of the somatic mutations that are present in the tumor cells. So we also talked about whole genome, whole exome. So what we do really a lot here at the New York Genome Center is whole genome sequencing, which really is trying to sequence telomere to telomere. We take all of the chromosome, we chop that into small pieces, we sequence with small reads, we align to the genome, and we get the entire coverage from one telomere to the other telomere for every chromosome in the genome. We can also do whole exome sequencing, so we have a technique for which we enrich for the region of the genome that code for the genes that are producing protein, and that's about 1.5% of the genome. We can also do panel sequencing, so we select a set of regions that is of interest, and so that can be a set of variants that are known and that we want to study, or that can be a fairly large collection of genes. And by large, in the cancer genomic community, I'm talking about a few hundred genes. We know those genes, we know what we can do about it, we know their clinical actionability. We want to study that in all of our samples, all our patients, and so that's what is going to be deployed. We also do RNA-seq, in which case we sequence the transcriptome. We sequence the expression of the genes at the RNA level. So this has been ongoing for 10 years, 15 years. They are large efforts. These are two large research efforts. The TCGA, the Cancer Genome Atlas, had the goal of sequencing the most frequent cancer type. And so they did whole exome sequencing and RNA sequencing for now about 20,000 patients. The International Cancer Genome Consortium did the same with whole genome sequencing. So again, take the most common cancer type, select some patients, apply whole genome sequencing, and share the data for the somatic landscape of the mutations in those common cancers. There are also clinical efforts done at the hospital, for instance, at Memorial Sloan Kettering, where they developed a test called MSK Impact. So they have a selection of 500 genes, and they try to apply that to most of their patients, and they collect the information for those genes that they know are of interest for their practice. There is also commercial entities. One of them is Foundation Medicine that offers similar tests, and that doctors can order and can get their patients tested on this test. And finally, there is now an AACR consortium that try to aggregate panel information from different hospitals that put together the information and the clinical information from those panel testing. And so now it's about 50,000 patients or profiles are hosted in this large database. So we did that for about a decade. We have somatic landscape of the most prevalent cancer type, and we have database and interface, and we have tools that we can use, we can study, we can look at what has been done, what do we know about somatic landscape in different cancer type. However, a big problem is that 75% of the patients are coming from Caucasian patients. And so this is here the results in TCGA, that's self-declaration. you see that about 80% of the patients in TCGA are European or from a European ancestry. This is in AACR Gini, and again, that's 76% here of self-declaration is white. Okay, that's a huge problem because I showed you we have some molecular differences between the ethnicities. So the goal of Polyethnic 1000 is to try to offer proactively, try to recruit patients that are non-white, try to induce some diversity in the public database. And by doing that, we will address several important questions about the role of ethnicity in oncology and offer genomic service to those patients from underserved population. So for that, we need to develop an infrastructure that will collect patients from a lot of different places that are serving those populations. And we will generate a research platform that will help us to understand the genomic determinant of ethnicity. We'll study the germline predisposition of specific cancer and define novel ethnicity-specific mechanism and biomarker of therapeutic responsiveness. Okay. So by doing that, we hope to have a platform that will serve regionally, nationally, and internationally for collaboration in data analysis and disease mechanism. So you've seen this map before. So we historically worked a lot with the institutional founding member of the New York Genome Center. Most of them are located in Manhattan. For this particular project, we decided to work with a lot of other institutions that are mostly in Brooklyn, in Queens, and in the Bronx. And you see here from this map that they serve a population that is very different than the population that is being served in Manhattan. So we have 16 sites for sample collection, 36 collaborators. We established some working group to really set up this infrastructure. And we have site coordinators and pathologists on site that help us to select the patient, select the samples, send them to us, and we will do the sequencing here. We'll do the analysis here, and we'll share all of the data with our consortium and progressively with the rest of the larger scientific community. The test we are going to apply is whole exome sequencing and RNA-seq. So the results are going to be the germline and the somatic single nucleotide variants, indel, copy number variant. We will detect fusion genes in the RNA-seq. So those are molecules formed by two genes that are normally not expressed together. We will identify expression outlier compared to external cohort, and we will be able to validate the variants that we discovered in whole exome by looking at their sequence in the RNA-seq. So this is detail of the pipeline that we will apply for the whole exome. I'm not going to go into detail. But on top of that, we will do ethnicity estimation. So by having the exome, we can look at what we call ancestry-informed markers and detect what is the genetic ancestry of these patients. And so that's the very classical test that you can see in the news. Ancestry.com or 23andMe is offering you something similar, where you spit in a tube, they will test some markers, and they will tell you that your family comes from Ireland or from Wales or from Germany or from elsewhere. But mostly it works very well with European ancestry. So by having exome, we have a lot more information than what they collect in those services, and we can look molecularly what is the genetic history of these patients. And that may be different from the self-declaration, from how people perceive themselves, because ancestry information reflects a much bigger time frame in the history of the family of these patients. We will also be able to look at HLA typing, so that's looking at the genes that call for, so the HLA genes, one of them was mentioned by Neville a little bit earlier, and we really believe that there is a lot of differences between the population that are related to the HLA genes. That is a very polymorphic gene. And so by having a clean definition of the HLA allele in each of the patients, we hope to discover some differences that may be interesting for our project. So we set up the project in 3 different phases. The first one is a feasibility study where we collect retrospective samples, 100 samples. We will do whole exome on the tumor and the RNA. We hope to move quickly to a second phase that will be prospective, where we will have patients fully consented for data sharing, where we'll do whole exome, RNA-seq. We hope to get about 500 or 1,000 samples from the only eligibility criteria is non-white patients. So people that self-declare as non-white are eligible for this study. And in the end, we hope to have an infrastructure by which we will be able to collect cohort. and say someone comes with an hypothesis of a molecular difference between Filipinos that have a lung cancer of a certain age, but they're struggling to collect samples because of their criteria, we can leverage our infrastructure and collect those patients from anywhere in New York. And we really imagine that in New York we have representative of every population on Earth. And so we really have the ability to put together the force from the different hospitals and collect or constitute large cohorts that will enable those studies. So here is the really preliminary results of this, and that's a very recent slide. Those are the first 24 samples that we collected. And you see that we have 15 patients that are Black or African American. We have 9 of them that are Asians. We have 7 men, 17 female patients, and we have a diversity of cancer type. That's really the first samples that we receive. We're going to sequence them, and we hope to be done with this first phase by the summer to have 100 samples, and then move to a second phase where we will have a retrospective study. One thing I did not mention is that in the retrospective study, we imagine that in some percentage of the patients, we will be able to discover an information that is clinically actionable. So this study is done in the research setting, so we are not allowed to return information to the physician or to the patient, but we believe that if we discover something that might be clinically actionable, we can order a confirmation test from a CLIA-approved laboratory and validate our finding, confirm our finding, and then transmit this information to the physician so that some patients can benefit from their participation in the study, and we can have a portion of the patients will benefit by the test that we're doing. So this is the large group of people that are involved in our steering committee and in the project, but also in all of the working group that are really helping us to set this together. The funding currently comes from the Mark Foundation and the Zuckerman Fund. And that's what I had to present to you today. And we have, there's a nice map. I don't know if it's represented very well, but that's what it is.
I should, before I take questions, emphasize that the long-term goal is to provide a test that can be used for treatment for all these patients. This is a gradual process. You can imagine how complex it is to have IRB and consent forms from all the hospitals that actually have never had genomic tests before, to get those in place, to have everything consistent, and then following all the rules, provide a test that can be actionable. And that's clearly our long-term objective. One of the important activities at the Genome Center is in technology development. And there have been extraordinary advances in the last several years on the speed of sequencing, the volume of sequencing, and perhaps most dramatically is in being able to look at individual cells, doing single-cell sequencing, single-cell analysis to look at different cell types. For example, there's a major effort internationally in the Human Cell Atlas is understanding the diversity and nature of single cells in different cell types, liver, heart, and so on. And we have a small but very effective and outstanding innovation group here. And Dr. Eleni Mimitou is going to summarize a very interesting aspect of the work there. So Dr. Mimitou is a senior research scientist in the Technology Innovation Lab, which, as I indicated, is a multidisciplinary team that really is approaching many different aspects of technology. Mimi joined the New York Genome Center in 2017, has been working closely with Peter Smybert, who's the manager of the Technology Innovation Lab. And the lab develops novel tools for genomics, and recently has focused on this problem of single-cell genomics with an emphasis of tools on obtaining multiple modalities of information from high-throughput single-cell assays. And that's what he's going to talk to you about today, is using an amazing set of tools to look at individual cells from both a protein and RNA analysis basis. So Dr. Mimitou was previously a research scientist at Memorial Sloan Kettering Center, and she's a very important part of our innovation team.
I'm very happy to be here representing our group, and I'm going to give you a very glimpse of the technologies we have developed the last couple of years, pertaining, as Tom said, single cell, trying to get as many information as we can from single cells. Site-seq and cell hashing has initiated a couple of years ago under the guidance of Peter Smybert and Marlon Stockius, another senior scientist in the lab. And so when I joined, I started working on extensions of that technology, which I will be touched on later during the talk. Allow me to take quite a few steps back just to make sure we're all on the same page and remind you how DNA stores and transmits its information. One of the major jobs that DNA has to do, the genome has to do, is to faithfully maintain and transmit the information, and it does so by replicating during its cell division. But also it needs a way of transmitting this information, and it does so by converting itself into a related molecule and a messenger RNA in a process called transcription. This messenger RNA is then translated into proteins, which do the majority of the work in the cell. They provide structure, they make energy, communicate. They are the cell's workforce. Now, when we talk about RNA-seq, it's a technology that allows us to profile those RNA, the transcriptome of the cell, the collection of the RNAs it expresses. Not as Neville touched in his talk, only a small portion of the DNA is actually used to transmit information. So there are genes, there are loci in the genome that are transcribed, the genes, and give those RNA molecules. When we want to get a glimpse of what genes are expressed in its cell, we sequence the RNA. Unfortunately, the sequences we have to date can only read DNA. So we have to convert RNA to DNA in a process called reverse transcription. And then we can sequence those molecules. Traditionally, and back when RNA-seq was introduced, it was performed in bulk. That means that cells coming from either homogeneous or heterogeneous samples were lysed, and RNA was prepared en masse. And then that was used to create complementary cDNA via the reverse transcription I mentioned, and get an average gene expression from all the cells. Unfortunately, this method doesn't allow us to address the cellular heterogeneity. Lots of the tissues that we deal with are very heterogeneous. Not every cell has the same expression profile. So with the advent of single-cell RNA-seq, this is not a problem anymore because we can get an expression profile from every single cell. And we can see that in, let's say, cell A, there's mostly that transcript. In cell B, there's these transcripts, and so on and so forth. So we can actually reveal heterogeneity and subpopulation expression variability in thousands of cells in parallel at the same time. I know this is a busy slide. I just want to draw your attention in the jumps in the throughput scale of single-cell RNA-seq that happened the last few years, and that's mainly due to the nanodroplet and pico-Weill technologies. We in the lab, we use extensively the 10x Genomics platform. It's a microfluidic device that allows formation of nanoliter size aqueous droplets in oil. And each one of these droplets can contain one cell and one bead. That means that in those thousands of droplets, we can partition cells and do thousands of different experiments in one tube. An analog that we often use to make the comparison between bulk and single-cell RNA-seq more relatable is that of a smoothie versus a fruit salad. In a smoothie, when you drink a smoothie, you get an average of all components, and it can be dominated by the most abundant type or the strongest flavor, fruit. On the other hand, when you eat a fruit salad, you can easily taste all the ingredients, even for foods that are not as flavorful or they're not in high proportions. So I hope I have convinced you the importance of doing single-cell RNA experiments when we care about the diversity of cells in our sample of interest. But how do we perform a single-cell RNA-seq experiment? The heart of single-cell RNA-seq lies in the formation of partitions where we have one cell and one what we call reverse transcription primer. This is usually delivered in a form of a bead. It's bound on a bead that can co-partition with one cell. What is special about these primers is that it carries a cell barcode. It's different for every partition, and it allows us to trace its transcript to a certain cell. It also contains an amplification handle, which is a sequence that is it's the same for all of these primers, and it only serves the purpose of making many copies of it so we can sequence it. And finally, it has what we call a poly T-tail, which acts as a magnet to recruit, to bind to all the mRNA molecules of the cell. Very conveniently, the mRNAs in the cell end in a poly A-tail, and that can bind which the poly T primer can bind to, and during reverse transcription in the droplet, it can copy the information of the transcript right adjacent to the poly A-tail. So now in one molecule, continuous molecule, we have the transcript information and the cell barcode information. We can do that for every single molecule during the experiment, and eventually we build, we collapse reads that have the same barcode, and we can assign all these transcripts back to the cell they were coming from by sequencing this, this part of the molecule. Now, before single-cell RNA-seq, there were other methods that researchers could use to interrogate complex cell populations, including flow cytometry. In flow cytometry, instead of RNA, you actually detect a protein. And you do so by using antibodies that bind specifically to that protein. Now, flow cytometry doesn't have, of course, the high throughput that RNA-seq does, but it has the benefit that you're detecting a molecule that is more abundant, the protein, and it can be more sensitive. So instead of having researchers decide whether they're going to use single-cell RNA-seq or flow cytometry to interrogate their cell populations, we asked, why can't we do both? Why can we detect from every single cell both the transcriptome and the protein, the surface protein? The challenge here was to make an antibody, which is a protein, sequenceable by a sequencer. If you remember from the central dogma of biology, we cannot go from protein to DNA. So plan B was to actually dress the antibody like an mRNA. We conjugate oligos that have a poly A-tail to those antibodies, and they can be incorporated in any single-cell RNA-seq workflow that is based on the oligo-DT priming. Moreover, upstream of that poly A-tail is a barcode sequence that that acts as an identifier of the antibody it's bound to. So for every different antibody, we have a different barcode. This is the simple yet elegant idea behind CytSeq. It stands for Cellular Indexing of Transcriptomes and Epitopes by Sequencing. And to take you quickly through a CytSeq experiment, the cells that we want to take through the single-cell RNA-seq workflow, before we do that, we stain them with the antibodies of interest. We wash them to remove all the unbound antibody. And then, here depicted is a microfluidic-based workflow. We bring cells and beads together in those droplets. Inside the droplets, the cells lyse, so every mRNA and oligotag are now released. They are poly A-tailed, so they bind to the poly T primer that is bound on the beads. And during reverse transcription, the information of either the transcript or the oligotag is copied into the primer that has the cell barcode information. So eventually we can make libraries that either give us information about the transcriptome or the oligotags present in the experiment. To see how CytSeq performs, we did a proof of principle experiment using blood immune cells that are well characterized and have highly heterogeneous cell populations. We stained with a panel of 13 antibodies and took them through the 10x chromium device. Commonly, in a single-cell RNA-seq, data are presented in the so-called t-SNE plots. In very simple terms, with t-SNE plots, highly dimensional data, such as gene expression profiles from thousands of cells, are reduced to only 2 dimensions for representation purposes. So cells that are similar, cells that have similar expression profiles tend to cluster closer together and form those islands with different colors, and cells that are different are further apart. So here we wanted to see how the data we get from mRNA sequence, if it correlates with the data we get from protein sequence. And again, in the interest of time, I'm only going to show you results for 2 markers, CD3 that stains T cells and CD19 that is present in B cells. And these are transcripts as detected in this experiment. When we look at protein, we see the very similar result. CD3 antibody stains this cluster, and CD19 antibody clearly stains that cluster. In fact, the staining is even, the counts that we get for antibody tags is much more abundant, confirming that detecting protein is a more sensitive process than detecting transcripts. But we developed CytSeq not as a way to confirm single-cell RNA-seq. We wanted to add on it. And in a series of experiments that unfortunately I don't have the time to show, we see that when we integrate clustering, multimodal clustering, so clustering based on both transcript and protein data, we get much better separation of the different cells than with a single-cell RNA-seq alone. As you can see here on the left-hand side, it's again immune blood cells. And you can see that just getting RNA information sometimes doesn't do such a great job of separating different cells. But when we get protein and RNA, the separation is much better. So we are widely using now this method with labs within and outside the New York Genome Center. And we extended this method to cell hashing, which is essentially CytSeq with antibodies against ubiquitous markers. For example, let's say you have 3 samples that you want to take them through a single-cell RNA-seq experiment, but you have money only for 1 reaction. You could easily label each one of these samples with an antibody that is differentially barcoded, and then pull these cells together and perform one experiment, one single-cell RNA-seq experiment. By using the barcode information from the antibody that was bound to cells before pooling, we can trace the cells back to the sample that they originated from. So we can either increase the number of cells that we can interrogate, or we can pool many samples together and save a lot of money. Towards the end of the talk, I want to talk about some additional CytSeq extensions that I was more involved into developing, which is capture of non-poly A transcripts, such as guide RNAs. As Neville mentioned, there's no way of overstating the importance of CRISPR screens. And recently, single-cell RNA-seq has been used as a readout for screens. Instead of having a biased output like survival or resistance to a drug or fitness, we can actually just sequence the RNA of the cells to infer how the perturbation mediated its effect. And unfortunately, the guide RNAs that guide the Cas9 into the target to perturb to the double-strand break don't have structure that is anywhere similar to mRNA. So they cannot be captured with the traditional single-cell RNA workflows. Instead, they have a variable region at the 5' end, which is the one that binds, recognizes the target site. And they have an invariable region of about 18 nucleotides it's called the scaffold. Now, recently, 10x Genomics released a product. It is different from the traditional in a sense that the primer that begins the barcoding process is now soluble instead of bound on the beads. In the original design, this primer is bound here and has the cell barcodes. In this solution, the RT primer is soluble, and that gives us flexibility and ability to customize what kind of transcripts we can capture. So we reasoned, why not, in addition to the poly-DT primer that is part of the kit, why not add a primer that anneals to the scaffold of the guide RNAs and incorporate guide RNA detection in the single-cell RNA-seq workflow. So in this case, this primer will reverse transcribe, and in parallel with the rest of the mRNAs of the cell, the complementary strand will obtain the same barcode that the mRNAs will. So from the same cell, we can get information about the transcript and the guide and the protein. To see how that performs, we did a simple experiment where we used a small guide library with targeting and non-targeting guides and asked, first of all, what's the capture rate? For how many cells can we get guide information? And we were really happy to see that the capture rate was very high. Only 1.7, for 1.7 cells, we could not detect guides. For 85, we could get 1 guide per cell, and for about 13%, more or equal to 2 guides per cell. Importantly, I'm showing you the result only for one guide here. When we target ITGB1, it's a gene encoding for CD29, a cell surface protein. Here we cluster based on guide information. So these two clusters have guides for ITGB1. And you can see that we have loss of signal at the mRNA level for these two guides and also at the protein level. So we can now use mRNA and protein as readouts to assess how efficient our targeting was, our editing was. And it's a great tool for people that want to do CRISPR pool screens. We termed this technology ExCyTeSeq, which stands for Expanded CRISPR-Compatible CyTeSeq. And it allows us to capture from single cells at least 5 modalities. mRNAs with the ability to do selective enrichment for transcripts of interest, such as T-cell receptors and B-cell receptors. And unfortunately, I didn't have time to go into this. Antibody tags, be either CyTeSeq for phenotyping or cell hashing for multiplexing, as well as guide RNA molecules for CRISPR screens. So to summarize, integrated protein and RNA measurements enhance cellular phenotype. Cell hashing allows multiplexing and cell overloading. Both technologies are compatible with both 10x technologies, either 5' or 3'. And finally, ExCyTeSeq allows the single cell capture of at least 5 modalities in a single assay, including direct guide capture for multimodal CRISPR screens. At this point, I would like to thank everybody in the Technology Innovation Lab. It's really a fantastic group to work with. Peter Smybert is the manager, and Marlon has been the spearheaded the CyTeSeq technology.
I should emphasize that a very important part of this that Eleni referred to is the coordinate development of mathematical tools to be able to interpret this data. And Rahul Satija, one of our faculty members, is one of the leaders in this area of really being able to take complex data sets like this and do the kinds of distributions that allow you to see patterns and to see how cells differ. And this is actually taking another step that one of the technologies that used here is called spatial transcriptomics. You can now take a piece of tissue, like a slice from a tumor, and you can visualize in the microscope where all the RNAs are being expressed. And it's a way of looking at the complexity of gene expression over the entire tumor sample. That allows you to begin to look at the evolution of tumors as the cells divide. So I hope you got a good flavor of what we do here. As you can see, it goes from computational biology to technology development, ultimately impacts on human disease, and that's what we do. I'd like to thank you all for coming, and we'll take any questions if you have any. Well, thank you.